Okay, now we're going to talk on carbohydrates. Carbohydrate, hydrate carbon. The monomer form of the carbohydrate is I'm using, like in math, CX2XO8. This would be an empirical formula. So for every carbon, there's a water. For every carbon, there's a water. That's where the term carbohydrate came from in the monomer. Carbohydrates, some of them are sweet to the taste. They can be called your sugars. Others that are not sweet to the taste can be called your starches. If I use my little symbology up here, the value X in the above formula can go from 3 to 7. That would be 3 carbons. This was naturally occurring. 3 carbons, 4 carbons, 5, 6. You can have trios, 3 carbs, tetros, pentos, hexos, and heptos. Most abundant are the 3 and the 5. Okay. The 3 are found in metabolic pathways that you'll see later in this course and in ANP2. And the 5, which would be ribose and deoxyribose, DNA, RNA, are found in DNA and RNA. So the 6th one, which would be like glucose, you would think is the most abundant, but not really, because you find more of 3 and 5s if you look in the human body. The carbohydrates make up about 1 to 4% of your cell mass. Now, if I took the 70 kilogram person, about 56 to 60% is water. That would be the water part, the liquid part. Then I'm into the solid part. And of the solid part, 1 to 4% are carbohydrates. 1 to 4%. Okay. Here would be some examples of trios, pentos, hexos that I mentioned before. Okay, that you can just glance at. I mentioned also that carbohydrates can exist in a linear form, 3-dimensionally, and a cyclical. And they prefer a cyclical, but they will oscillate between the linear and this. This form is the most thermodynamically, remember thermodynamics, energy stable in the universe. Okay. When we look at bonding carbohydrate monomers together, the bonding occurs, if you recall, by removal of water, a dehydration synthesis. So we would be going from the monomer to the polymer, I told you before that organic chemistry is covalently bonded. So that bond would be a covalent bond between them. But the bond would be what we'll call a glycosidic bond. We'll call this, quote, a glycosidic bond, right there. Okay? The glycosidic bond, that would be only used to hook monosaccharides together to form a polysaccharide. Terminology. When 2 monosaccharides are hooked together, we call it a disaccharide. When 3 to 10 are hooked together, we call it an oligo. Now, this is by good etiquette. Good etiquette means, like, if you go to a nice restaurant, you could eat the whole meal with your salad fork. Is that good etiquette? No. So good etiquette would say if you have 2 together, disaccharide, 3 to 10, oligo. Oligo in Latin means few. As I mentioned before, knowing a little Latin sure helps in clinical situations. And more than 10 is called a polysaccharide. Now, that's by good etiquette. Some books you'll see a 3 and they'll call it a poly. That's not the best etiquette, but it's okay. Okay? When properly identifying the glycosidic bond, just like in the big world, shape is important. If you had a Volkswagen being tugged by an 18-wheeler, then the chain between the two would have a certain 3-dimensional orientation. I'll say it like that. Well, bonds also have certain angulations. They have certain angulations because they are a 3-dimensional entity, much smaller than we can see. So there are 2 really different shapes of glycosidic bonds. The alpha bond and the beta. The alpha and the beta. So when I examine disaccharides, the most common disaccharides that we would have, which would be sugars, remember, they would be sweet, an alpha-1,2 linkage. Now the alpha-1,2 linkage would basically be between the first carbon, remember we talked about number, the first carbon of one and the fourth of another, or the second of another. It just depends which one. Of the first of one to the second, in some cases the first to the fourth. I'll show you that in a moment. So an alpha 1,2 linkage is table sugar. An alpha 1,2 linkage. An alpha 1,2 linkage between a glucose and a fructose. An alpha 1,4 linkage, common in beer and so forth, is maltose, which is between a glucose and another glucose. And then a beta 1,4 linkage is between glucose and galactose, which forms lactose, which is in milk. You know, you've heard of lactose intolerance. So just showing you some things. So right here would be showing you glucose to glucose, which forms maltose. Maltose, alpha 1,4. Right here is glucose to fructose. Glucose to fructose, which is sucrose that you see right there. Sucrose, a 1,2 linkage right there. Okay. So this just shows the alpha and beta glucose ring structures that we can have. Okay, so common disaccharides that you have I'd like you to be familiar is sucrose, which is table sugar, which is glucose bonded to fructose. In your metabolism, if you're doing AMP2, you break down when you come down after you digest it and bring it in, you break the glucose from the fructose and work on them separately. Maltose, glucose to glucose. Lactose, galactose to glucose. See, lactose intolerance is where you don't have the enzymes that can break down lactose. Enzymes have to break these bonds. So that lactose will go undigested through your intestinal tract into your large intestine where resident bacteria exist. And they love that lactose. And that's what gives the diarrhea and the gas and everything. So again, here are some common disaccharides that you encounter. Okay. We then quickly go to oligosaccharides. Oligosaccharides are 3 to 10. I will talk more about them when we talk about the cell membrane, because on the cell membrane, oligosaccharides exist, and they determine some of the specificities of your cell, identification markers. We will talk about that at another time. Okay. We now go to polysaccharides. Remember I said 10 or greater. The polysaccharides generally are not sweet. Therefore, they're called your starches, and you've heard of that in the food industry. Some are called fiber. Fiber are polysaccharides that your intestine cannot digest, called cellulose. Because it cannot digest it, it adds what we call bulk to the diet. You may have heard of Metamucil and things of this nature. They would be fiber. The purpose of polysaccharides is either for storage of energy. Now, you know we got plants and animals. We store our carbohydrates inside cells. Animals do, not just us, as glycogen. Plants also need storage. So they store it as something called amylose and amylopectin. Carbohydrates, polysaccharides also give structure. So we have this cellulose I mentioned, which is fiber. Then we have something called chitin. I'm not sure how many of you have heard of the arthropods and the crustaceans, that shell that they have, and some on some of the insects, that hard shell. That actually is chitin. It's a complex polysaccharide. Okay. So here's amylose, which is stored in plants for energy. Here would be a bunch of glucoses, alpha 1,4 together, long chains of glucoses. We actually break that down when we eat a plant. Okay. Here's amylopectin. It's another one. It's more of a branched unit. We won't get too much into branching since I'm not doing, but that would be alpha 1,4s with some side groups of alpha 1,6s. But again, that's for plants. Okay. I come to glycogen. Glycogen is how we store. It's animal starch. That's how we store our energy inside our cells. And when we need energy, we're using carbs. then we'll reach into the cell, break a little glucose off from the glycogen, and burn it. Because remember, we don't have to eat 24/7. Now, bacteria do. They have to be around their food all the time. So here would be glycogen. It's heavily branched, a bunch of alpha-1,4s, and then that's that 6-carbon, alpha-1,6. So that's what causes this branching. It gives a nice structure to store something in a small space, rather than just a long chain being folded. Okay? Fiber is a beta-1,4 link. Now here's the situation on fiber. Cellulose, a plant has a cell membrane and a cell wall. We said again, a plant has a cell membrane and outside of that a cell wall. Animals just have a cell membrane. The cell wall of a plant acts as its skeleton. That's why when the plant doesn't have enough water, it wilts, because the cell itself is inside this skeleton, this cell wall. And it's kind of like a balloon in a box. The balloon gets deflated and the box caves in. If you inflate the balloon, the blocks will cave out and get strong. So cellulose, though, cannot be digested by us as humans because we don't have the enzymes. So it goes undigested and it gives bulk to the diet. It makes the intestinal muscle have to work against it, just like we lift weights to keep our muscles strong. That's what we call cellulose. If you look at it and tease it out, it has a fibrous appearance. That's where the term fiber came in, a fibrous appearance. Why can't we digest it? Because we don't have enzymes to break down this beta-1,4 linkage to break apart the glucose molecules. But cows do. That's why cows can eat grass, and some other animals. Dogs, to a certain extent, have minimal. That's why when they get sick, they'll start eating grass and so forth. Okay, so there's that. Okay, chitin. Chitin is that hard stuff, as I mentioned, in the crustacean and so forth, the exoskeleton of arthropods and crustaceans. That's a complex polysaccharide. But what have we done? We've been able to take that chitin and utilize it to make certain suture materials in medicine. So see, we take benefits from these various things there. So that gives you somewhat of an explanation on carbohydrates, starches, sugars, sweet to the taste. So what I'd like for you to do is review my PowerPoints and also read it in the book, because these are very important things. Many of you will have to take a nutrition course. We'll also do a little nutrition A&P too. So start glancing at that. I hope this helps. And I'll also probably have a video too that's not just mine to also explain this. Thank you very much.